The ASV portal is found at asv-portal.biodiversitydata.se, and it's an interface to sequence-based observations in SBDI. As such, it provides two entry points for searching using sequence alignment or filtering, as well as an option to submit metabarcoding data to the ASV database and the SBDI bioatlas. And we'll start in that end, so I'll just click submit to go to the submit page. Here we list the main steps of the submission process. The first of which is to submit your raw sequence data to the European Nucleotide Archive, the ENA, and we provide a guide to do this, so we'll just have a quick look at that. Our guide to ENA submission is currently at GitHub, and obviously the ENA provides a lot of documentation already, but we thought users would benefit from a step-by-step guide with examples specifically for metabarcoding data. So you can go through these steps and have a look at our examples, and hopefully you will find that useful. Now we'll just go back to the ASV portal submit page. So after submitting your raw data, the next step is to denoise your data into amplicon sequence variants. And as Daniel just showed you, you can use the AmpliSec pipeline for this. Step number 3 is then to download, fill in and upload the data input template. And before we have a look at this template file, I should say that the Living Atlas platform and GBIF, the Global Biodiversity Information Facility, uses the Darwin Core format. And we largely follow this in the ASV portal, of course, but we do add a few specific fields for processing data in the ASV database before they go into the bioatlas. And we can now have a look at the template file, which I put up here. So this is a spreadsheet file with 6 tabs or sheets, and 5 of these are for data entry and 1 is a guide sheet. And inside this guide sheet, there is a gray section, which is basically a list of fields as they appear in the data sheets and some notes on the sheets themselves. Then the green section is the original Darwin Core format definitions, comments and examples. And then we added a yellow section, which contains specific interpretations for metabarcoding data when applicable. So if we go back to the top left corner here, we can read that the event sheet mainly contains fields derived from Darwin Core categories, event, location and material sample. And this just means that the event sheet contains information about when, where and how you collected your material sample. Then we have the DNA sheet, and that one contains fields mainly derived from the Darwin Core extension called DNA derived data. And this is the extension that was recently developed by GBIF in collaboration with us and our Scandinavian and Australian colleagues. And it is also in part derived from the Genomic Standards Consortium's MIX checklist, and therefore the fields correspond to the fields reported to the ENA as well. Next up is the EMOF sheet, and EMOF has fields derived from the Darwin Core extension called the Extended Measurement of Fact, and that is what you use for contextual data. And depending on how much detail you want to provide on units and methods and so on, you can actually opt for using the EMOF simple instead, and we explain this in the yellow section. So finally, we have the ASV table sheet, and that is a pivot or cross tab of the Darwin Core occurrence class fields. And this just means that if we have a look at that, we have the ASVs as rows and then some data on their sequences and taxonomy. And then you have the samples as column headers, and these values here are the sequence reads. And I think that is pretty much a typical output for bioinformatic pipelines, so it should be straightforward for you to fill in. Right, so once you have filled in your data, you return to the submit page, and you can just click on the upload button. So if you're not logged in already, this will take you to the regular BioAtlas login page. And if you don't have an account already, you can sign up using this link. And even if you do have an account, if this is the first time you submit data to the ASV portal, you do need to request upload permission, and you will be taken to a page that tells you how to do that. My account already has upload permission, so I can just log in, and that will take me to the upload page. And here I can just select an Excel or archive file of a maximum size of 800 megabytes and then upload this. Right. Yes, and then we can go back to the submit data page. So once you have uploaded your data, it just says that once we have looked at the data, we will contact you and to write a data sharing contract, and your data will then be published in the ASV database and BioAtlas. So that was all I had to say about submitting data to us. And now we can go back to the start page and have a look at the options for finding ASVs and associated BioAtlas records. So we'll start with doing a search based on sequence comparison using the basic local alignment search tool, or BLAST. So click BLAST here, which takes me to the BLAST form. And this is where you can paste in your own sequences that you want to align with the sequences in our ASV database. And in BLAST terminology, your sequences are called query sequences, and the sequences in our database are called subject sequences. And the pasted sequences need to be in FASTA format, and we'll just use the default sequences here. The next two fields are used to set two basic parameters for the BLAST algorithm. And these are the minimum identity and the minimum query coverage. So if you only want to find exact matches, you can use the defaults. And these mean that you require the full length of your sequences to be aligned, and that all of these aligned bases need to be perfectly matched between query and subject. But actually, the beauty of BLAST is that you can tweak these thresholds to find related sequences as well. So we can, for instance, set this identity to, say, 93% instead. And then I can just scroll down and press BLAST. And this takes us to the BLAST result table. Right, and this table then shows in the bottom here that we have 20 matching ASVs, and the table shows these in batches of 10. You can, for instance, click and show individual sequences, and you can also download BLAST results directly by selecting them here and select a format. We can have a look at one of those result files here. The important thing that I wanted to show is that if you want to link data in this downloaded file to some bioatlas records later, this is the field that is sort of the common denominator, and that is the taxon ID. Right, so we can go back, and there is also the option to go directly to view the bioatlas records, but we're not going to do that right now, because we are going to look at the other search option, which is the filter search, and we can use this top menu to go there. And this takes us to the filter search page, and the available search filters are grouped into sequencing details or taxonomy related items. And the selected filters are combined with logical and limit available options in the remaining drop-downs. At the moment, we only have a single ASV dataset in the bioatlas, so sequencing options are obviously limited here, but you can select the 16S rRNA gene for prokaryotes and combine that with genus. And I'm going to go with the Flavobacterium here. Then I click filter. And before we have a look at the results, I should just say that the prokaryote taxonomy is a work in progress for both SBDI and GBIF, and we actually use the GBIF taxonomy backbone in the bioatlas. And at the moment, this means that there are some discrepancies between the taxonomy in the ASV portal and in the bioatlas. But importantly, the taxon IDs, which I just showed you, these are derived from the actual sequences, and they are consistent between the two systems. So again, when we run this search, we get a result table, and we can have a look at sequences if you want to. And we can download data as before. But now we want to have a look at a specific ASV here and look at the corresponding records that are currently in the bioatlas. So I just click Show Bioatlas Records, and this takes us to the occurrence records page in the bioatlas. And apparently there are 15 occurrences of this specific ASV. You can, for instance, have a look at the map here to look at the geographic distribution of this ASV. And so we see that these are bacteria occurring in the Baltic Sea. You can go back to the records and then look at a specific record. Right, so the occurrence record of an ASV looks pretty much like a regular record page in the bioatlas, but it does have some characteristics that are specific to ASV data. So I'm going to point to a few of these. First of all, we can have a look at the previous identifications. So this is a field that we use for showing the taxonomy as it was reported by the data provider. And this can then be contrasted with the taxonomy section here, which can differ because this is the taxonomy that SBDI has assigned to this ASV using a standard method and a reference database for a specific group of organisms. And we can also go back up here and look at the identification references. So if you click this link, you will get some more information about how we perform this taxonomic re-annotation in SBDI. And you can also look at the identification remarks, which summarizes this taxonomic annotation and shows you the confidence for this specific ASV. Right. So we can also go back to this field here, which says, no, sorry, here, the organism quantity type. So this says how we have quantified this organism, and this value should also should always be DNA sequence reads for ASVs. And the organism quantity, the actual value, is shown here as 165 reads then, obviously, for this ASV. But in metabarcoding, you will typically want to relate this to total read count. So then you have to scroll down to the additional properties down here. And here it says sample size unit is also DNA sequence reads, and the sample size value is this. And in close proximity here, you can also see the material sample ID. So this is the biosample ID for the corresponding raw data at ENA. And one final thing here is this field. So most of the sequence related information is packaged into this new Darwin Core extension that I told you about before for DNA derived data. And this has not yet been implemented in fully in the Living Atlas platform, which means that to make sure that all of these data are available to users, we have also added these as so-called JSON strings under this dynamic properties header. So that's the reason behind that. And so finally, now that you are in the BioAtlas, if you want to search for specific ASVs directly from within the BioAtlas, that is not via the ASV portal, you can also look at the top menu and go to Search and Explore, Search Records. And there's a tab here called the Batch Taxon ID Search. So if I am correct here, we should be able to just paste this ASV ID or taxon ID in here and do a search, and that will take us back to the same result page as before. Right, so that was all I had to say about the ASV portal right now. Thank you.